guys, so let's do chapter 8. Here are the sections. There are 5 of them. I'm going to try to get through at least sections 1 and 2 in this video. Maybe section 3. We'll see how it goes. Section 1, cellular respiration. It is a cellular process, that's in the name, that breaks down nutrient molecules produced by photosynthesizers. Those are sugar molecules. And they produce ATP. It consumes oxygen and produces carbon dioxide. It is an aerobic process, which means that it requires oxygen. It usually involves the complete breakdown of a glucose molecule into carbon dioxide and water. Energy is extracted from the glucose molecule in a stepwise release process, and it allows ATP to be produced very efficiently. It involves oxidation reduction enzymes that include NAD+ and FAD as coenzymes, so they are parts of the enzymes that make these reactions happen. They are very important. As you will remember, most chemical processes would not occur without some kind of enzyme facilitating it. Here we have the cellular respiration equation. We start with a glucose. We add 6 oxygen molecules. The oxygen is reduced to form water, and the glucose is oxidized to form carbon dioxide. You'll notice that no matter is being created or destroyed in this equation. Okay, we start with 6 carbon, we end with 6 carbon. We start with 12 hydrogen, we end with 12 hydrogen. We start with 18 oxygen, we end with 18 oxygen. In cellular respiration, electrons are removed from substrates and received by oxygen, which combines with a proton with a hydrogen ion to become water. The glucose is oxidized and the oxygen is reduced. Here we have a leopard of some kind, a zoomed in view of its cell, and an even further zoomed in view of the mitochondria. This is where most cellular respiration occurs. So we are making ATP from ADP. So the whole process of cellular respiration, the goal is to stick as many phosphates on as many ADP molecules as we can. So you may be wondering from the previous slide, what is NAD+? Well, it is, and I'm probably going to pronounce this wrong, nicotinamide adenine dinucleotide. It is a coenzyme of oxidation reduction. Okay, it is oxidized when it gives up electrons and reduced when it accepts them. And these NAD+ molecules are used over and over and over again, just like every other enzyme. They are not used up in the reaction. FAD is another one of these enzymes. It's flavin adenine dinucleotide. It's also a coenzyme of oxidation reduction, and it's sometimes used instead of a NAD+. It accepts 2 electrons and 2 hydrogen ions to form FADH2. It can accept 2 hydrogens because it has accepted 2 electrons, and that negative charge of the electrons is attracted to the positive of the hydrogen. So if I have 2 negative charges, I can attract 2 hydrogen ions. Phases of cellular respiration. Okay, it includes 4 phases. Okay, glycolysis is the breakdown of a glucose molecule into 2 molecules called pyruvate. Okay, this happens in the cytoplasm, and it basically just snaps that glucose molecule in half. And it produces a small amount of ATP. And it does not use oxygen, which is an anaerobic process. Now, all cells go through this glycolysis, okay? But only eukaryotic cells, plant and animal cells, can go further than the glycolysis. Okay, so you might have been wondering, Ferris, if we have a bacteria cell that doesn't have a mitochondria, how does it get its energy? Well, it gets it from this process called glycolysis. Our second phase is the preparatory phase, or the prep reaction. Both molecules of pyruvate, that glucose that we snapped in half, are oxidized and enter the matrix of the mitochondria. So if we go back to our diagram up here, the matrix is this inner space here. Let's see, electrons store energy. Electron energy is stored in the NADH, so it gains a hydrogen ion. Okay, and 2 carbons are released as carbon dioxide. We get 1 CO2 from each of those pyruvates. So we've snapped it in half, and then we take a carbon off of each one. The next phase is the citric acid cycle. This occurs in the matrix of the mitochondria, and it produces more of that NADH and FADH2. It is a series of reactions that releases 4 carbons as carbon dioxide, and this cycle happens twice per glucose molecule, once for each pyruvate. So we snapped it in half, we took a carbon off of each, and now we're taking it through that citric acid cycle. It produces 2 intermediate ATP molecules per glucose molecule. And then finally, the last step where most of the energy is generated is the electron transport chain. It's very similar to the electron transport chain in photosynthesis. Okay, it extracts energy from those NADHs and FADH2s that we have made, and it passes electrons from higher to lower energy states. It takes that energy and sticks the phosphate onto ADP, making it into ATP. Now, if it's really, really, really efficient and absolutely nothing goes wrong, this electron transport chain can make up to 34 molecules of ATP. So if we add in the 2 that we net in glycolysis, each glucose molecule can generate 36 ATP molecules. So here is a diagram of the 4 phases. Okay, so out here in the cytoplasm, we have glycolysis. We break that glucose molecule into 2 pyruvate molecules, and then it is transferred into the matrix of the mitochondria. Then the prep reaction happens. Okay, that's whenever we're making the NADHs and the FADH2s. Then we get the citric acid cycle, where we are further breaking down that pyruvate molecule and releasing carbon dioxide. And we're making even more NADH and FADH2s. And then finally, we take the energy from those intermediate molecules and we make a lot of ATP in the electron transport chain. Okay, section 2. We'll see if we can get all of this done before I run out of time on this video. Outside the mitochondria, glycolysis. Glycolysis occurs in the cytoplasm outside the mitochondria. It consists of a series of 10 reactions, each one with its own enzyme. There's an energy investment step where 2 ATPs are used to activate that glucose and split it into 2. So we've used 2 ATP molecules. But don't worry, we're going to generate 4 of them in this glycolysis, but we only end up with a net gain of 2 because you got to put 2 in to get 4 out. The energy harvesting step, the oxidation of G3P, remember that molecule from photosynthesis, it occurs by the removal of electrons and hydrogen ions. 2 electrons and 1 hydrogen ion are accepted by NAD+, resulting in 2 molecules of NADH. 4 ATP are produced by substrate-level phosphorylation. Okay, this is when an enzyme passes high-energy phosphate molecules to adenosine diphosphate. Remember that unenergized one? It sticks that phosphate onto it, making ATP. So there's a net gain of 2 ATP because we started with 2 that we had to use. So that's an investment of 2. We made 4, so we have only netted 2 ATP. Both G3Ps are then converted to pyruvates. And those are the molecules that are going to go into the mitochondria. So here we just have a visual representation. Okay, our inputs are 6 carbons in the form of glucose. We input 2 NAD+ molecules and 2 ATP molecules and 4 ADP and 4 phosphates. Okay, we have as our outputs 2 pyruvates. Each one contains 3 carbon molecules. We now have 2 NADH molecules, 2 ADPs because we used them to get the reaction started, but we've also stuck these phosphates onto the ADP gaining us 4 ATP, but we have to pay back the 2 that we borrowed before, so we end up with a net gain of 2 ATPs. So here we have our 6-carbon chain, our glucose. It is split using those 2 ATP molecules. It is split into G3P, that's the molecule that's made during photosynthesis. Okay, and then we oxidize it. The oxidation of G3P occurs as NAD+ receives high energy electrons. And then we get some substrate level ATP synthesis whenever we stick that phosphate onto the ATP. And we also end up with a 3-carbon chain with a phosphate at the end. And there we go. We end up with 2 molecules of pyruvate that can then go into the mitochondria. Here is a diagram showing how the enzyme actually puts the phosphate onto the ATP molecule. And I don't think we're going to have time for section 3, guys, so I'll put that in the next video.